MES Marampally e-learning.
Hello everyone, this is Rubina, Assistant Professor, Department of Biosciences, MES College, Marampally. Today, I will be dealing with the topic Staphylococcus.
Staphylococcus is a round-shaped bacteria that constitutes the normal flora of our skin, but they can cause opportunistic infections also. Coming to the introduction, all gram-positive cocci are divided into two groups based on the catalase test, that is, their ability to produce catalase enzyme. Catalase positive microorganisms or catalase positive cocci comes under Micrococci, and catalase negative cocci comes under Streptococci family. Micrococci in turn consists of 4 genera, that is Micrococcus, Planococcus, Stomatococcus and Staphylococcus. Likewise, Streptococci consist of Streptococcus, Pneumococcus and Enterococcus. In this video, we will learn about Staphylococcus. Coming to the characteristics of Staphylococcus, they are gram-positive organisms, that is, they give purple color after gram staining. Here we can see the purple colored gram-positive Staphylococcus. They are arranged in clusters and they are non-motile, non-sporing, facultative anaerobes, and they may be capsulated or non-capsulated. Based on the coagulase test, they are divided into two. Coagulase positive Staphylococcus aureus. There is only one type of coagulase positive Staphylococcus, and that is Staphylococcus aureus. And the second group is coagulase negative Staphylococcus or cons. Example, Staphylococcus epidermidis. In the coming slides, we will be learning about Staphylococcus aureus in detail. Staphylococcus aureus. They are seen as bunch of grapes and they give golden yellow colonies in nutrient agar. That is why they are given the name aureus, which means golden. Here we can see the golden colored colonies on the nutrient agar plate. In McConkey agar, they appear as pink colonies as they can ferment lactose. Here we can see the pink colonies in this figure. They exhibit hemolysis on blood agar, which is evident by the presence of clear zones around the colonies. Now we will learn about the virulence factors in Staphylococcus aureus. They are cell wall associated factors, toxins, extracellular, enzymes. First, we will see the cell wall associated factors. Cell wall associated factors consist of peptidoglycan, teichoic acid, cell surface adhesins, protein A and microcapsule. Coming to the peptidoglycan, they have thicker peptidoglycan layer and it gives the strength and rigidity to the cell wall. The second one is teichoic acid. It helps in the adhesion and prevents opsonization. Next one is cell surface adhesins. It constitutes of clumping factors which forms the basis for the coagulase test. The next one is protein A, which helps in the inhibition of opsonization and cause platelet damage. Finally, the microcapsule, which inhibits phagocytosis. Coming to the second virulence factors, that is toxins. Toxins consist of hemolysins, alpha, beta, gamma and delta. Hemolysins, they cause hemolysis. Then the second types of toxins is leukocidin. Leukocidin exhibits the leukocidal action, that is, they helps in killing leukocytes. The next toxin is exfoliative toxin, which cause staphylococcal scalded skin syndrome, SSSS. The next toxin is enterotoxin, which is involved in food poisoning. The last type of toxin, it is toxic shock syndrome toxin, that is TSST. It leads to the multi-organ damage and is caused by poor menstrual hygiene. Third and the final virulence factors are extracellular enzymes, which consists of coagulase, staphylokinase, heat stable thermonuclease, deoxyribonuclease, protease, hyaluronidase, lipases and phospholipases. Coagulase enzyme is involved in the blood coagulation, which forms the basis of coagulase test to differentiate coagulase positive Staphylococcus aureus and coagulase negative Staphylococcus or cons. Staphylokinase, it is responsible for the lysis of fibrin clots in blood. Heat stable thermonuclease and deoxyribonuclease is present only in Staphylococcus aureus. And the next one, protease, it helps in the lysis of proteins. Hyaluronidase helps in the lysis of connective tissue network. Lipase and phospholipases helps in the fat breakdown. Coming to the pathogenesis of Staphylococcus aureus, it starts with the colonization at skin folds, usually at axilla, perineum, nose. Then it enters the body through cuts or aberrations, followed by invasion using enzymes such as proteases, hyaluronidases, etc. Then they evade the host immunity by producing various toxins. Finally, they spread to various organs and cause several clinical manifestations. The clinical manifestations include skin and soft tissue infections, that is furuncles and carbuncles. Musculoskeletal infections, usually osteomyelitis. Respiratory tract infections, that is ventilator associated pneumonia. Bacteremia, that is sepsis and septic shock. Urinary tract infections. Toxin mediated illness, TSS, that is toxic shock syndrome, and food poisoning. The lab diagnosis of this Staphylococcus infection staphylococcal infection includes sample collection and the gram staining of this sample collected, then culturing, microscopy, biochemical tests, antimicrobial susceptibility tests of the sample collected. Now we'll discuss about coagulase negative staphylococcus or cons. Cons includes Staphylococcus epidermidis, which cause prosthetic devices related infections. Staphylococcus saprophyticus, which cause UTI, that is urinary tract infections. And Staphylococcus lugdunensis, which cause endocarditis and osteomyelitis. Thus, we have learned about the general characters, peculiar features, virulence factors, pathogenesis, clinical manifestations, and lab diagnosis of Staphylococcus. Hope you got an idea about this bacteria Staphylococcus. So, thank you all. Thank you for watching. Thank you.